The mass spectrometry data was analysed with two software, Peak Studio and MaxQuant. These assigned PSMs from database searching against the E. coli K12 genome. This was followed by quantification. Assigned protein groups from the Peaks database search were passed through PeaksQ, a built-in quantification tool in the Peaks platform. The protein groups from MaxQuant analysis were passed through LFQ Analyst, an online open access quantification tool originally designed for MaxQuant outputs. Both of the quantification outputs were then visualized with the string database extension for Cytoscape. The outputs from both platforms' quantifications are typically a list of protein accession IDs, associated confidence scores, and denotation of changes in abundance across treatment and control samples. In order to understand the relationships between these listed proteins, services such as String will scour publications to calculate correlations algorithmically. Cytoscape is an open source platform for visualizing protein interaction networks, which can be imported directly from String, or when using the String extension, can be queried in the Cytoscape interface. String assigns confidence scores to protein interactions based on lab experiments, genomic context predictions, co-expression of proteins, algorithmic text mining, and co-occurrence of proteins, as well as providing publications from Uniprot, PubMed, and others for further reading. String scores can give a useful indication for the potential relationships between proteins, but must be followed up on with further reading, as they don't provide a directional basis of the relationship. I generally gave the text mining scores the least value, as they are based on the presence of gene names on the same documents. However, many of the protein interactions given by String have only text mining scores, so further reading of publications can determine whether these relationships are valid. I found Cytoscape useful for the edge table, which can be seen at the bottom here. It lists scores for each protein interaction with the total score and constituent scores easily viewable. It also allowed direct importing of tabulated mass spectrometry data and direct exports of network images and score tables. Selecting a node will give the Uniprot description of that protein and show a diagram of the structure. I also found the enrichment function in Cytoscape easier to use than String. You can select one or several of the proteins in the network, then click Enrich Publications to obtain a list of publications used to calculate the String scores, or click Functional Enrichment for a list of Go biological pathway publications. Here are some examples of the enriched publications at the top and the functional enrichment publications at the bottom. These will list the number of genes involved, as well as the background genes, the confidence score, and the publication names and PubMed access IDs. From both outputs, I obtained a list of 29 proteins identified to change in abundance. Inputting these two lists into Cytoscape gave the two networks that can be seen here. Because of the variability in the outputs from LFQ Analyst, I generally had more confidence in the proteins listed from the Peaks quantification. There was only one protein identified in both quantifications, LAPA, also known as YCIS, a lipopolysaccharide assembly protein. It was shown to decrease in abundance by effectively the same amount across both platforms. This heat map shows proteins increasing in abundance in red and proteins decreasing in green. Only 4 increased, all of which were only identified by Peaks. I began by investigating these 4: TIG, RLPA, WRBA, and RPLC. TIG was found to be more than double the abundance in the treatment sample against the control. String showed that this protein interacted with DNAK, another chaperone protein involved in chromosomal DNA replication, which was about half as abundant in the treatment samples. String gave this interaction a high score, so I began to read publications which I used to infer that the increase of TIG proteins and decrease of DNAK proteins was in response to the ampicillin, potentially indicating that the treatment E. coli was downregulating the genes involved in cell replication and upregulating TIG in order to remove aggregated proteins being either directly or indirectly inhibited by the beta-lactam antibiotics. Further evidence for me was the 5-fold decrease of HOLA in the treatment samples, which is a key enzyme in bacterial DNA replication, showing suppression of replication mechanisms. Because beta-lactam antibiotics inhibit the cross-linking of peptidoglycan, the increase in abundance of RLPA, which is involved in peptidoglycan binding and maintenance of rod structure, was proposed to be an attempt for the treatment E. coli to utilize a differing pathway for peptidoglycan linking away from transpeptidases. RLPA is also involved in recycling of peptidoglycan monomers. Through reading of enrichment publications, I found that WRBA plays a role in biofilm formation. Bacteria will form biofilms in order to protect themselves by limiting penetration of ultraviolet radiation, oxidative stress, drugs, and temperature fluctuations. The image shown here is from a publication by Rossi et al. Showing the difference in biofilm formation from a regular E. coli and a WRBA deleted mutant. This publication identified that WRBA deleted mutants form thinner biofilms than their normal counterparts. I proposed that this increase in WRBA proteins gave evidence for the formation of biofilm as well as the presence of RAPA peptides in all samples, which is a protein identified to be present in biofilms but not their associated planktonic cells. In bacterial antibiotic resistance, the formation of biofilm is generally coupled with increased expression of efflux pump genes. Many of the proteins involved in efflux pumps were found to actually decrease in abundance, but none were deemed statistically significant by either quantification platform. As the typical method of bacterial beta-lactam resistance is the expression of beta-lactamases, which were not found to be present in any sample, I proposed that the increase in biofilm-associated proteins, the decrease in DNA replication and cell division proteins, and increase of proteins potentially utilising a different pathway for peptidoglycan linking were the general mechanisms of the treatment E. coli's ampicillin resistance.